Okay, Nick, so we're here at the Wolfville site, where it's our first stop in our field trip today. So what do we got planned in here?
Well, it's like this lawn. If we wanted to sample this lawn, we could do random numbers, and we could grid the lawn, and we could do random samples. But a quick and dirty way would be to run a transect across it, and then go at predetermined spaces. It could be every 5 meters, but you want to do something that is not subjective and that you can compare from site to site. So we're going to be running a tape measure as our transect, determine a start and finish point, and then in addition we can put a quadrant together, and this is a very standard thing in ecology, and we can get some percent cover data, and we can also look at abundance of things that we might not even be thinking of. When we go out here, we're looking at a natural grass community, that's a domesticated grass community, and we can look at spiders, we can look at other invertebrates, amphipods. We don't know what we're going to see, but we don't want to miss the opportunity to get an insight into some part of the food web.
Sounds good. All right, so let's make our way out to the site.
So we're here on the dikes. This is the modern dike. It probably parallels an old Acadian dike. We can see over here to our seaward side that we've got another dike system, and we're going to be going further along to another dike system. And I think what the Acadians would have done was that they would have started diking a portion of land, and then they'd extend it, and they'd extend it until the original dikes were inland, and their perimeter dikes would be out. So it would be a whole sort of system. And Nelson and I were talking about how certain animals do this. You know, the foraminifera, they might make a small shell and then increase the width and increase the size of the shell, and moving out of one house and into the other. This was how the Acadians brought more and more land into agricultural production. You can see that the modern dikes, that face wall is a rock dike, and we were talking about how what the Acadians did was much more efficient. They used the dike grass itself. Remember the salt hay? They used the salt hay itself to make the dike. They didn't have bulldozers, they didn't have the rocks, they made it out of itself, much more efficient.
So Nick, in terms of a place to sample, we're going to walk out this way onto what would have been some of the original dike, and you think this might potentially be a more nutrient poor sort of area?
I think this would be more nutrient poor than the sewage treatment plant. So right now we're going to go down and we're going to start to see how this operates. So right now, interestingly, this is the low nutrient system, and you see how this is a much finer grass. That's a much finer grass than the flatter grass that was the disticus. So this is a mat, and what we were saying today was all of this amphibod. Little amphibod.
Is that a mud snail?
It's a little crustacean, flattened crustacean.
Okay.
So here's our thatch, and remember the competition is all below ground. We start here on the seaside and in the low channel areas. We have that plant that is so much a colonizer, and it's the only plant that can do the real dirty work, the sort of super inundated conditions. remember, it can take waterlogging, it can take seawater, salinity, and what we're talking about. Imagine these plants, even at the low tide that we're at right now, these plants are breathing. Remember, the oxygen is going into the leaves, straight down into the stems, and going through the rhizome system and getting to the roots. And also remember that the plant is an aerobic organism and that it has to have oxygen throughout its tissues. And this is a big stressor of most plants, but Spartina alterniflora, the cordgrass, can grow. So if we start at this colonizing plant that's stress tolerant, and then imagine that the sediments are built up, and there's more deposition of organic material, and we're moving in time as a marsh develops from mud, moving in time, and finally we get to an elevation where we get a grass starting. And this grass, we're leaving behind Spartina alterniflora cordgrass, and we start to get this grass that is more competitive. This is salt hay. This is the salt hay the Acadians would have used, and they would have dug sods with this and laid them on top of one another. And we'll show you the original dikes.
Great. It's good the students are out here today because my fingers are going numb. It's so cold.
Oh yeah.
Thanks. We're going to use a quadrat system here.
So we're mixing the two systems that we talked about. We've got a transect, and we're saying this is about 200 meters. We're going to want some data, so we're going to take 5 plots between here and the sea edge, because we want to understand how, what the composition of this high marsh is. This is a quadrat. A quadrat is a 4-sided thing. This is a meter squared quadrant. Good that you can just pull this thing apart and put it back together. So we're putting it together, and there we go.
So the quadrat is just for making sure you work within a defined area.
It's to give us it's a defined area, and in this case we're going to get percent cover of plants in a meter squared. It's very standard, and it's very nice that people believe, ecologists, that we're giving a visual estimate, we're saying it's quantitative, and there's a lot of trust involved. So have a look at this quadrant. This is all dead material from last year. This is last year's growth. This is Spartina patens, the salt hay, and here we have some Spartina alterniflora, the cordgrass. This is a better competitor. Remember what it's competing for. It's competing for below ground resources. This is a better stress tolerator, but it can't stand the competition from the root system of this plant. Now, if we look at it from the top, we estimate that this plant covers most of the area, and one could say that there's 95% cover of Spartina patens, and this is a waterproof book. It's already getting muddy. So what day is it? It's May the 1st, 2021, with Nelson O'Driscoll, and the course, we are at Wolfville Harbor, and Quadrant 1, Transect 1, and we said salt hay 95%, and we've got this broadleaf here. This is called cordgrass. What do we think this is? The way I do this is I try and move the cover into into one of the quarters of the quadrant. So I'm moving this cover in, I'm moving this cover in. I think I can fit all of these, all of this cover of the broadleaf into 25% and maybe less than that. So I'm going to say 18%. I do this repeatedly, and so the error I have here, it balances out over the whole transect. Now, we said also we want to take these data because there might be stuff in here that we don't know that's important, but it could be. Are there any invertebrates here? There's a little amphipod. See that little, who's here? Spider.
Lots of little critters living under the grass.
And so if you were here, you'd be counting these. We could count, but we don't have time to count, but we're just going to note that we have amphipods and spiders.
Great. So for lining up with tomorrow's activity, where we're looking at the distribution of mercury and in the sediments, this site here would be called WOLF Wolf 1-10, I think would line up with. So I'll make sure that we coordinate between those two.
So we're at position 2 in the transect, and we have the same constituents. But don't worry that it's boring, but that you're laying down some data, you're laying down some documentation that this marsh is pretty much dominated by one super competitor, Spartina patens. We see where it gets lower. We have Spartina alterniflora, the plant that can take the waterlogged conditions, but where the elevation's a little bit higher, we have Spartina patens, the salt hay. Remember, it's winter, and in summer this whole place is green. This is last year's growth. We do the same thing, quadrant number 2. Spartina, and I should have said that really these things overlap, and we change that 95% to 100% because if you look from the top, there's no mud sticking out, and so that's 100% plant cover. And underneath this Spartina alterniflora, that broad grass, is more of the fine grass. The fine grass being Spartina patens. So Spartina patens again 100%, but we have a little bit here. I try and move it into one of the quadrants of the quadrant. So this is a 25% area, and it's obviously not 25%. And then we say, no, it's obviously not 12%. It could be, if we move it all, it could be 6%, let's say 5%.
So it's a best judgment estimation of how much is
best
patens.
judgment, and believe it or not, this is how a lot of plant ecology is done. When you're doing a plot that you're coming back to year after year, you get very, very adept at making good decisions and trying to narrow that down to your best estimate, because you're looking for year-to-year differences.
Good.
Okay.
Some more amphipods here. Some little
wrigglies.
yeah.
Did you catch the wriggles, or did they wriggle away?
I think they wriggled away before I got them.
Okay, we'll get
Yeah.
that. Yeah, amphipods. Here we are, quadrant 3. We look down. We're seeing essentially the same as we saw last time, and we put that in place. We've got 100% Spartina patens and a little bit here, and that is, let's say that's 1% Spartina alterniflora, cordgrass.
So you're just looking for the broad-leafed grasses in there and estimating.
Yeah.
So a little tiny bit over there on that edge.
Yeah. Ooh, we got wrigglers. Nelson, did you get the wrigglers? There they are, by my pencil. 2 little amphipods.
Okay. Who's that guy?
Yeah. Good. We're again on transect 1, quadrant 4. Looking down, we don't have 100% cover. We see mud here, so we don't have 100% cover. We try and put that mud together. It's much less than 25% of the quadrant. I estimate that to be 10% of the quadrant is mud. So we say 90% is Spartina patens salty hay. We look down, do we see any Spartina alterniflora? No, it's missing here. This is all dominated by one grass. Very low diversity, very high productivity. This is the summer growth starting. You see the summer growth, the green shoots of Spartina patens, and that's going to produce the same kind of above ground thatch that last year produced, and then this stuff will slowly get ripped up by the tides, and that will go into the ocean and sustain the food web. No amphipods seen. It's a cold day. Transect 1, quadrant 4. We're looking down at percent cover. What do you see? You're seeing a thatch, and we're estimating cover from thatch, and we're realizing that there's layers and layers of plants, so you can have over 100% cover. But at the very least, you've got 100% cover of this fine grass, Spartina patens. So quadrant 5, Spartina patens, 100%. And now we're looking for the other grass, Spartina alterniflora. That's here. We move it all into what we would, yes, can easily do that. Moving this in here, estimating and thinking that I'm going to say that's 20% cover. Spartina alterniflora. Looking in at the mud, layers and layers. Oh, there's our little crustacean. There's another one that's a little bit too cold to move.
They get away so fast.
These big stems of Spartina alterniflora, and then the thatch. Okay, and this is the high marsh, but notice within this high marsh there are channels, little creeks, and the creeks start draining the marsh, and you go from the high marsh to the sides of these creeks where we see the cordgrass, Spartina alterniflora, and some people have been fertilizing experimentally these marshes, and when they fertilize the marsh, it changes the plant dominance from the very competitive at low nutrient Spartina patens. It changes it to other grasses that are not putting much into the below ground resources, so it sets up an erosion pattern. And what they'd found in Massachusetts was that it led to the erosion of the Saltmarsh if there was very high fertilization. University behind us. We've got Wolfville Harbor. You see that shine of the mud, and that mud is nicely rounded, convex humps. We're suggesting that the mud is held in place by the diatoms because it's actually more of a silt, and the ooze from the diatoms holds that mud together. We see the low area, which is Spartina alterniflora, coming up to the salt hay. The salt hay is all around us. There's a saltmarsh pond here, and that saltmarsh pond is interrupting the salt hay. All around the saltmarsh pond is Spartina alterniflora. It's now draining out because it's low tide. And then we have this feature that is a straight line. It's a straight line, and this is an old Acadian dike. We have a plant we haven't seen before called Iva, and this Iva, or sumpweed.
Sumpweed?
Iva frutescens.
Okay.
And you see this Iva is all along the old Acadian dike.
Why does it grow there in particular, Nick?
It grows here because it's more competitive than the Spartina patents. It's now we're moving from below ground competition to above ground competition, and this Iva is setting up. So we finished our first transect, and we look out here and we see the alternation of the high salt hay, and then we've got these channels, these lower depressions of Spartina alterniflora, the cordgrass. And one of our expectations is that as sea level rises, we're going to start to lose the salt hay regions if it can't keep up with this coastal squeeze. So far, we're suggesting that the salt marsh has been keeping up with sea level rise, and if it keeps up with sea level rise, we expect to see the salt hay. But then, as the water comes up, maybe a little too fast for that salt hay to keep putting on a new layer, we're going to see the transformation of the salt hay to the cordgrass. And we're also thinking that as the level of the sea has come up, that relative to the dike, the salt marsh keeps coming up and up and up and increasing in elevation. Now, on the other side of the dike, you know what we're going to expect. We're going to expect to see agricultural land. We look at the agricultural land, and this being the dike, a modern dike. This is not the Acadian dike. It's probably laid down on top of the Acadian dike. We look down here, and we see agricultural land, but we also see that the agricultural land, well, it looks like it's lower relatively in comparison with the dike surface than over here, where so far the level of the marsh has been keeping up. So we've got two possibilities. Either this side of the dike has slumped, but the marsh level has slumped. The marsh is now agricultural land. It's slumped, or maybe it's lost sediment because of erosion, or that this level has been rising up in relation to that, and that the old dike has stayed the same and is now agricultural land, whereas this has just been increasing, and we said that it's probably increased by 30 centimeters so far over this century.
So here we are at the outflow channel, kind of mid-tide, where the sewage is outflowing here. And at high tide, of course, this would be filled with water and mixing in over the land surface. Nick is just walking out towards our contaminated area, we're going to call this. This would be Wolfville 1-1 site.
member valve. It's not the wooden one we showed you in the presentation. That's pushing out a nutrient source. It's interesting here that you see that there are patches of green algae. We don't know what this green algae is, but it's likely reflecting the fact that there's an increased nutrient concentration in the waters. Now we come to these edges. It's a good site because we can see the difference between the rooting system of the two dominants. We've got Spartina alterniflora, and if we look down here, we can see the rhizomes, those big rhizomes that we talked about of Spartina alterniflora. Imagine that this rhizome is bringing oxygen down, 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 and then the oxygen goes through this rhizome system to these roots that come off the stem. If we broke that open, that's where you can see that's what's bringing down the oxygen. And then if you look in comparison with these, a much, much finer rooting system of Spartina patens, and here it's restricted to the top, and it's a much, much finer system. One of the questions that I have that we won't be able to answer, but it should be something that we keep in mind, is whether this calving that you see, calving I mean that the loss, the break off of the sod, whether this break off is due in part not just to the mechanical effects, but that with the nutrient, the more the plant gets nutrient, the less it has to invest in the long run to get the nutrients if there's free nutrients. So one of the hypotheses is that as the plant puts less into the rooting system, that the whole structure of the ecosystem, that the marsh, becomes more erodible. So that's something we should keep in the back of our mind. We can't say for sure, but that is a possibility and one reason that we want to reduce the eutrophication of these natural systems. So here we are. This is our second transect. This is our first transect at the nutrient site. And we look down and it's Groundhog Day again. We see Spartina patens. We don't see much else. One of the things we could look at here was saying, well, there's much less above ground material. That there's more mud. So if I look down at this and I just took a picture and was trying to say how much of that was vegetated, I would say, I'm going to say 85% Spartina and 15% is mud. There's no other vegetation in this quadrant. And do we have our friends, the amphipods? We've got something like a sea lavender that's just germinated here. No amphipods yet. There's some green algae. This is the green algae we saw by the river there. By the creek. There's a trace of green algae. So then we'll move on. Quadrant 2. Transect 2, essentially the same as we found last time. It's salt hay, and it's 97% salt hay, 3% mud, and nothing else. You look inside. We're not finding as much thatch here. The length of the grass seems to be smaller, seems to be shorter. And okay, there's our amphipod. He's gone. There's a spider. A little black spider. It's different from the first one. If we were doing invertebrates, we would be collecting these. And a portion of our diversity would be these spiders. There's our amphibod again. So the same sort of invertebrate community. And one of the things that Nelson and I were talking about was that perhaps this site is subject to more current more flow-through erosion, and so that would be a source of difference, a source of error. Okay, we should also point out how chopped up this landscape is. If we look out here and look at those islands, which are on the creek side coming out of the sewage treatment plant, how green they are. The greening up could very easily be related to nutrient coming from the sewage treatment plant. Quadrant 3, same thing. We're doing 25-meter distances between quadrants, so it means that we can't find what we might want to say is at the site because we're doing it systematically. And that's very important that you keep on with your plan, that you don't say, oh, we'd like to do it here where we have Spartina alternative flora. No, you do a certain number of samples until you get a reflection of the same thing, Spartina patens. And green algae. So what we'd be looking at here, if we were thinking that nutrient might be involved, we'd start to go into these filamentous green algae that you see at the base of the stems of this salt hay. Right here. And we're just in spring, it's very, very cold, but still we've got a growth of algae at the base of these plants. And that's not a bad thing, it's just a response to available nutrients. Quadrant 4 of this second site, and you can see that even though we've gone across a landscape that is of similar elevation, we're getting a shift in vegetation, and we're getting a shift so that we've got a mixture of the two grasses. We've got the fine salt hay, Spartina patens, and then we've got Spartina alterniflora, the gourd grass. We've also got patches of green algae at the base, and we're not interpreting at this point because we're taking data, but in the back of our mind we're thinking, is this going forward? That is this moving from Spartina alterniflora, gourd grass, to a higher elevation, salt hay, or is this a salt hay stand that has been inundated or fertilized so that the plant community changes? This is a transitional stage, and so it's either going forward in succession to a higher elevation system, or it's going back in succession to a more flooded system, or it could be that this is a result of increased nutrient, which means that the salt hay is no longer competitively dominant below ground. So this is our last quadrant in the second transect, and you see again that we're in a cordgrass dominated system. Last time we had what did we have? We had 25% salt hay and 60% cordgrass. Now we're looking at cordgrass, and yet we can see that there is some salt hay in this. And so I'm thinking that what we're seeing is a shift from we're seeing a shift from the salt hay community to the cordgrass community. And as we said last time, there's two reasons that this could be. It could be a nutrient effect, or it could be that it's sea level rise and that the tolerance of the salt hay is exceeded, because as we say, this cordgrass can be inundated like rice. It's got lovely arenchyma, whereas this salt hay usually gets wetted on only the highest tide cycles, and so it's intolerant of constant submergence. We're at the second site, the sewage treatment plant. We see the modern dike. The modern dike is made of rock and is built higher and higher as the level of the sea is rising. We see that this could be thought of as coastal squeeze, that we've got cordgrass, and cordgrass is replacing this salt hay, and this salt hay is up against a rock environment where it can't go higher than its current position, and so as the sea level rises, we may lose this salt hay community and be left with the cordgrass community. Up higher in the rocks, this is called litter. It's the plant debris from all these marshes. You can see it's composed of Spartina alterniflora. This is the cordgrass. And it's also composed of the finer Spartina patens, and we can see there's a bryozoan. That's a colonial invertebrate. There's more of the bryozoan, and all of this material gets broken up and moved into the bay, and this is ultimately the food source for the zooplankton in the Bay of Fundy. And this is called the litter layer. It also includes seeds of the plants, and so this is good for sort of recolonizing dead sites.
So here's the direct outflow from the settling ponds from the sewage treatment plant. And you definitely can smell it when the wind is blowing in the right direction here.
So this is a creek which is coming off farmland. It's draining the farmland on the high tide. You can see there's a green field there and a recently plowed field to our right, and that runoff goes through this little creek, comes down, and goes through this abattoir which has been modernized. The same thing with the flapper valve. We see there's a freshwater grass which is called reed canary grass, but then as we come up, we get a mixture of the salt hay. This, believe it or not, even though we're on the inland side of the dike, this is salt hay. I think we're going to recognize it now if Nelson comes real close. We put a quadrant down here. We say, yes, this is the same grass as we had on the other side. Now, this is salt hay. We come up through the salt hay, and we have bits of Spartina. We have bits of the cordgrass, and now we come into. We have the green algae. This is cordgrass, Spartina. with me?
Yep.
The only place where we see cattails is right at the edge of the total slope of the farmer's field. Now, cast back to the video that Nelson took of the creek which was running out of the sewage treatment plant. The sides of that were totally dominated by the cattail. So we're seeing a more healthy, lower nutrient runoff of these farmers' fields than we are the high nutrient sewage treatment plant. As one
video here, this is just showing the farmer's field across from the dikes. Yeah. It's a good example of being able to see, don't fall in the water there, Nick. Being able to see the change in redox pattern. You can see the reddish iron oxides towards the top and the iron sulfides towards the bottom there with the grayish black change.
Wow, Nelson! This is just super cool.
What are you seeing?
So what I'm seeing here is these are old periwinkles from the marine phase of this area. These are. Can you focus on this?
Yep.
So these are old periwinkles. That there is a ribbed mussel shell right there. These are periwinkles, ribbed mussels, and then as we go along, we're getting this really low chroma, this graying out of the iron oxide. Tells you that from here down, this is a wetland soil. We're getting iron deposited on the outsides of these rooting systems. Maybe some of this is the old cordgrass, but essentially this is what we were talking about in lecture, how the agricultural land is borrowed from marine environment.
You can see where they flipped it up on top of the soil here, the gray patches from the anoxic zones.
So we are at the south end of Long Island, and Long Island used to be an island between offshore of the mainland. This is Grand and between Grand Prairie, the mainland, and Long Island is a large area of Saltmarsh, which you will see on Google Earth, which is all farmland. We're looking at a beach area which the locals call Sunset Beach, and the low area is, as you'd expect, it's cordgrass. We rise up in elevation, and right now we're going to sample what is pretty much a pristine salt marsh. It will be better flushed than the last we saw, so it will have a lower nutrient content. We see a farm over here, the green farm, but there's a buffer area here. You see that the salt marsh is actually coming into a woods area, so we're not up against a bank. We're looking at the most original salt marsh that we've visited yet. So we're going to do a transit this way. We've got a lot of room on our hands, and we want to sample it. We'll do every 50 meters. First quadrant. This is our first quadrant in this low nutrient system, and you can see here it looks very, very different from the cordgrass sites, or rather the salt hay sites. The tops of the vegetation have been washed off over winter, and what we're looking at is the juncus Girardi, Juncus Girardi, or it's also called black grass, but it's not a grass, it's that lily that I was telling you about. There's a small plant called Potentilla argentea. It's in the rose family, yellow flowers, and it looks to me that this is, here is Spartina patens, this long grass growing with the Juncus. So what we were saying earlier was that this is the better competitor below ground, but when you apply nutrients or when nutrient level increases, it reverts to Spartina patens. People have done lab experiments on this, and it's difficult to mimic it, but in the field, this is what people find. Transect, pristine farm site, quadrant 2, and it's Spartina patens 100%. And we have amphipods at the bottom of this oak, and just one sprig, so we'll call that a trace, one sprig of Spartina alterniflora, which is the cordgrass trace. So we are at our low nutrient site, and we found a pattern that we didn't expect. We're finding that in addition to Spartina patens, we have this grass which has flatter leaves, and this is Disticlus, the alkali grass. Reasons: there could be creek coming through here. We have a lot of water. We have a farmer's field, and this could be a response to a nutrient inflow from a field. And so finding the differences between these nutrient levels, think about how we might else design an experiment like this. Okay, so the alkali grass was restricted to an outflow of this farmer's field, and now it seems like we're back to more of a low nutrient system. This is the only salt marsh I know which is bordered in shrubs rather than a dike system. And so now we have ocean, salt marsh, and where you see those paper birch, that's a swamp meaning another wetland, which is the wetland we're in, which is the salt marsh system. We have both of the other high marsh species here. I've just found washed in. This is the black grass, and you see here what we're looking at is individual capsules, which, as I said in lecture, they remind me of Easter lilies. They don't remind you of Easter lilies, but they tell me this is not a grass. This is related to the lilies. It's called Juncus Girardi, or black grass. This is our last site for the low nutrient site. There's a very healthy crop of Spartina patens, and Spartina patens, as you know, is salt marsh hay. Very good. As I was saying to Nelson, I like this salt marsh because the salt marsh grades into shrubland. Some of this is shrub swamp, but it gives you the idea that as sea level rises, this salt marsh can progress inland and not get squeezed. This is the only place so far that we visited that the salt marsh will not get squeezed. We have the salt hay, and now the salt hay grades into a freshwater sedge association. This is a sedge called Carex recta. So we've moved salinity. We're getting some freshwater influence from the shrub zone, and we keep going with the sedges. And now we've got a freshwater grass. You can't really identify at this time of year into roses, but you can see that in another 50 years, this area could be the salt hay zone where the trees could die and the whole system can move inland. So there's no coastal squeeze, and this is the kind of planning that we need to do for our salt marshes. Of time. We don't know how old this is, but this is from when the salt marsh was more of a freshwater system. Here's a tree. These tubers, they could possibly be that Carex recta that we saw. And so now we're going through this phase, and this is being replaced by more modern salt marsh, and now this is being replaced by Spartina alterniflora. This is the big rhizome that you know. This is the cordgrass, and then now we're into the finer material, and we get on top, and what we see is it looks like, and it's now giving way to, it's being shorn off, and this whole system is calving down. But you see, we've got the upper salt marsh here. This upper salt marsh has gone through a cordgrass phase. So this is modern times. We don't see any cordgrass up here, but we do see cordgrass here, meaning that the succession went freshwater to cordgrass to salt hay. So we're in Port Williams. This is the mouth of the Jijiwaktuk River. You see that the river is actually flowing out in this direction. But if we look with the camera, you can see that the river now seems to be going upstream because the tide is coming in. We still have maybe 5 meters of tide to come. And we're looking at the bank of the river. It's an estuarine situation. It's saline. It's never fresh water. And this is cordgrass, Spartina alterniflora. And now, what do we expect when we come up and we see that the cordgrass grass is replaced by a more competitive grass? And what we're seeing here, and what you may start to recognize, we're seeing a completely different grass, and you see these spikes. This is a spike grass called the alkali grass, which is a grass that is less competitive at low nitrogen concentrations. So our hypothesis was that perhaps we would see a change in the plant community, but we're also aware that there are a lot of other factors. And one of the factors we saw over at our pristine site was that when things get wet or when there's a localized runoff from a farmer's field, we can get the same sorts of signals. Here we're also looking at an old bridge. You see across the river there's a platform, a wooden platform beside the bridge. That's where the old Port Williams Bridge came, and it would have come somewhere in the vicinity of where we're standing. So that's a disturbance factor. So when we're doing ecological research, we need to be aware of all these potentially confounding environmental impacts. One, mouth of the Gigiwaktuk River. And we will say, we're looking at the flat grass being disticlous. We're looking to see if there's any, yes there is, there's some salt hay. So we have a mixture of salt hay and disticlus. The question we're asking ourselves is this a transition? Is this a stable situation? Are we getting salt hay being replaced by the disticlus as nutrient contents come up? We should also show that this is a very different situation because we're on both sides of where our transect is going to go. We're getting Iva frutescens, this sort of shrubby herbaceous plant. Route 2, mouth of the Gigiwaktuk River. We're seeing the same mixture of alkali grass with little bits, and it's very difficult to do quantitative measurements here. I'm just going to say 50-50, as in the last stage. We had our amphipods in the last quadrant. We're spacing these quadrants at 25 meters. We're also writing ourselves a rule that if we hit a wet area, we're going to put the quadrant to a drier edge of that wet area. So we're trying to avoid that confounding effect of moisture. So this is our third quadrant. We're rushing through them. If you had more time, how would you design this experiment? If you had more resources, how would you design this experiment? This is one of your to-dos for Nelson and my project. And if we look at the quadrant, there's clearly a lot more of that flattened grass, which is the disticlus or alkali grass. It's mostly alkali grass, and we have to look hard for any Spartina patens in this quadrant. So we've got a replacement. If we believe the story, the hypothesis that we've laid out, we have a replacement of salt hay with disticlus. I'm gonna say there's a trace. Let's say there's 5% Spartina patens and 95% disticlus. So estimating this type of cover is difficult. If you were doing this and you wanted to be exact, you might take a sort of a sub area approach and say, okay, it's very difficult to tell overall how much disticlus, how much of this flattened grass you've got, and how much of the narrower grass you've got. You might then say, okay, I'm going to go to 5 areas, and here's an area, and what do I have? I have Spartina patens, I have Spartina patens, I have disticlus, I have disticlus, I have disticlus, and you can get a ratio and then multiply by your cover. If I'm looking at this, I'm going to guesstimate just based on informal assessments that I've got a 60% disticlus, 40% patens. We're in Port Williams. We're on our 5th quadrant, and we're calling this our nutrient-rich site because of the Gigiwaktuk River, which drains a lot of farmland and a lot of industry, and that nutrient comes down and is channeled through this narrow passage. It is a saline environment, it's an estuarine environment, and we're finding that the plant community is different. Is this just by chance? How many samples would you need to take? How would you take them? How would you sample this gradient? Here, we're looking at our last sample. We have some disticlus, and we've also got this shrubby herbaceous plant called Iva. We're looking up now where Iva comes from, whether it's coastal plain in origin, whether it's an exotic species, but it's very different. We saw it in Wolfville on the running dike, the old Acadian dike. Here we see a running path that might be a dike, but it's gotten off that and it's into the high marsh. But we didn't see that pattern in Wolfville. So here we have mostly Spartina patens, first in abundance. Second in abundance would be this clump. You would look and see how much cover that made up. Optimally, you'd want to be sampling this in June, July, when there was foliage. And then we have little bits of Disticlus. I'm going to say Spartina patens is 80%. I'm going to say that area there is mostly Iva. And I'm going to put that in for 5%. There are different layers. Sometimes in herbaceous communities you come up with 150% total cover. Okay, that's the end for today for our field work. We're going to get the data ready for you and we'll be back.